Welcome to the deep dive. Our mission today is to give you the ultimate shortcut to understanding one of the most exciting corners of modern biology, functional genomics.
It's a field with just incredible potential. We're talking about mapping entire regulatory networks inside our cells.
And really accelerating how we discover new drugs, right?
Yeah, exactly. But there's this immediate barrier, this huge wall called combinatorial complexity.
Okay, what does that mean in simple terms?
It means if you want to test the effect of, say, knocking out gene A, then gene B, then both together, the number of experiments you have to do just explodes. It's impossible to do it all in a lab.
Which is where the computers come in. These big deep learning models, things like GEARS and scGPT, are meant to solve that. They promise to predict the outcome for genetic changes that have, never even been tested.
It sounds like the perfect fix.
Hmm?
But that leads us to the central question we really have to dig into today.
Which is?
To what extent are these incredibly sophisticated models truly learning the specific biology of a gene? Or are they learning something else entirely?
And the source we've analyzed, a new framework called Systema, seems to have a pretty stark answer.
It delivers a major reality check. It shows that the performance metrics for these state-of-the-art models have been, well, consistently and pretty significantly overestimated because of these hidden biases in the data.
Okay, let's unpack that. This starts with what you called the shocking baseline revelation. So the standard task is this. You take a gene the model has never seen before, and you ask it to predict what happens to a cell when you knock it out. You're predicting that change in gene expression.
Yep. And to see how good the big models are, CPA, GEARS, all of them, the researchers pitted them against two almost laughably simple baselines.
These baselines are designed to be naive, right? They only capture the most general average effect.
Totally naive. They ignore all the specific biology.
We have to break these down because the fact that they work so well it's the whole story. So, first one. The perturbed mean.
The perturbed mean is the definition of simple. You take every single cell in your training data that's been perturbed in any way.
Knockout, overexpression, anything.
Anything. You just lump them all together and you calculate one single average gene expression profile.
And that one average, that's the prediction.
That's the prediction for every single new gene. It's a total one-size-fits-all guess. It has zero specific information.
It's basically saying, I don't know what this gene does, so I'll just guess it looks like an average messed up cell.
Exactly.
Okay, what about the second one? The matching mean for, say, knocking out two genes, X and Y, at once.
That one's a tiny bit smarter. It just takes the average effect of perturbing X alone and the average effect of perturbing Y alone and, averages them together.
But what if you've never seen gene X or gene Y before?
That's the key. If either one is unseen, you just swap it out for that super general perturbed mean we just talked about.
So we have these billion parameter deep learning networks on one side, and on the other, a simple average. It's like pitting a supercomputer against someone who just says, the average temperature on Earth is 15 degrees Celsius, and uses that for every single location.
That is a perfect analogy. And the unsettling part is, using the standard metrics, that simple guess performed comparably to, or sometimes even outperformed the supercomputer.
Give us the headline. Where did the simple average really shine?
For single gene predictions, the most common task, the one-size-fits-all perturbed mean baseline, actually beat every single sophisticated method on the Pearson score. Across all data sets.
It was the best performer.
It was number one. And get this, in the Norman 14 dataset for two gene combinations, the matching mean baseline beat the best deep learning model, GEARS, by an 11% margin.
11%. Okay, let's pause on that for a second. If a simple average of all perturbed cells is beating these advanced AIs, it means the complex models aren't learning the unique biology. They're exploiting some huge shared signal that's present across all the data.
You've hit it exactly.
Yeah.
They're taking the path of least resistance. And that path is paved by something we call systematic variation.
Okay, systematic variation. What is that, and why is it so easy for a model to exploit?
Systematic variation is just a consistent, widespread difference between all the perturbed cells on one hand and all the healthy control cells on the other. It's a background signal.
Like a stress response.
It can be. It could be from selection bias or confounding factors or, a general biological stress that happens no matter what gene you target. The models just learn to predict that general damaged state instead of the specific effect.
Let's talk about selection bias first. How does the choice of genes create this problem?
Well, think about older data sets like Adamson 13 or Norman 14. They didn't just pick genes at random. They picked genes that were all involved in, say, the cell cycle.
So they're all functionally related.
Right. And when you perturb a bunch of related genes, of course, their expression profiles are all going to shift away from the control cells in a similar structured way. The model learns that family resemblance, not the individual's identity.
That makes sense. But what about the really big modern screens? Surely they're less biased. Which brings us to this cell cycle story.
This is such a great example of a confounding variable. In the big repligal RPE1 screen, they found this massive systemic shift. They noticed that 46% of their perturbed cells were stuck in the G1 phase of the cell cycle.
46%. And the controls.
Only 25%. I mean, that difference is astronomically significant. There was some hidden biological mechanism at play.
And what was it? Why were they all getting stuck?
It's because the RPE1 cell line has a functional p53 pathway. Many of these genetic perturbations cause something called chromosomal instability, or CIN, and p53 is like the cell's emergency brake. When it detects that instability, it triggers cell cycle arrest in G1.
So the big signal the model sees isn't the unique function of the gene you knocked out. It's this giant flashing neon sign that just says cell cycle arrest.
That is exactly what systematic variation is in practice. The model learns the smartest strategy isn't to predict the subtle gene effect, but to just predict the massive cell cycle arrest signature, because that correlates with almost everything.
So how did the researchers actually quantify this? I know they used a geometric measure, this average cosine similarity. Can you walk us through that?
Sure. Imagine gene expression is a huge map. A cell is a point on the map. The effect of knocking out gene X is like an arrow, a vector, drawn from the average healthy cell to the average gene X knocked out cell.
Okay, so that arrow is the unique effect of gene X.
Right. Now, systematic variation is asking, how much does that specific arrow for gene X point in the same direction as the average arrow for all perturbations combined?
So if all the arrows are pointing in roughly the same direction, the systematic effect is dominating.
Precisely. If the cosine similarity is high, near 1, it means everyone's marching in the same direction. And in those biased data sets like Adamson and Norman, it was very high.
And here's the kicker, right? They found that this measure of bias correlated almost perfectly with the model's performance.
It's the smoking gun. For the Gears model, the correlation between the amount of systematic variation in a data set and getting a high performance score was 0.95.
Wow. A 0.95 correlation. That basically proves the old scoring system was broken. A high score didn't mean your model was smart, it just meant your data set was biased, and the model found the bias.
It was a measure of bias exploitation, not biological learning.
Okay, so we clearly needed a new rulebook. And this is where the Systema framework comes in, designed to neutralize this problem.
Right. Systema's core idea is to change the reference point. The old method always compared predictions to the control centroid, the average healthy cell.
Which created that problem of all the arrows pointing away from it in the same direction.
Exactly. Systema proposes a new reference, the perturbed centroid.
So the new 0 point is the average of all the perturbed cells combined. How does that fix things geometrically?
It's brilliant. You're essentially subtracting that universal stress signal from everything. You're no longer asking, how does this compare to a healthy cell? You're asking, how does the effect of gene X differ from the average perturbed state?
So you're forcing the model to find the unique signal.
And geometrically, it worked perfectly. The average cosine similarity, that measure of shared direction, plummeted from around 0.41 across data sets down to about negative 0.06. Basically 0. The shared directionality vanished.
Okay, so this is the ultimate reality check. What happens when you reevaluate the big, fancy models using this new unbiased reference point?
The scores fell off a cliff. They dropped substantially across the board. And crucially, those simple baselines we talked about, their scores dropped to near 0.
Which finally proves they were only capturing that systematic bias.
Right. Systema basically showed that predicting these specific unseen gene effects is, and I'm quoting, a remarkably challenging task, way harder than we thought. The new scores had a very low correlation with bias, just 0.21, proving the framework is robust.
So if the task is that hard and the scores are that low, does that mean these sophisticated models are useless? Do they capture any real biological information?
That's the next critical question. And to answer it, Systema uses another metric called centroid accuracy.
What's that?
It's basically an identity test. It asks, is your prediction for gene X actually closer to the real gene X data than it is to the data for gene Y or gene Z or any other gene?
So it's about distinguishability, not just correlation. And what did they find?
Overall, the scores were still pretty low, but one model stood out, a fine-tuned version of scGPT. And this is where we found a pocket of real biological insight.
Okay, so what did scGPT actually learn?
In one of the big screens, it was especially good at predicting the effects for genes involved in a core cellular process, translation, the genes that code for ribosomal proteins.
Ah, so it might struggle with unique genes, but it seems to have learned something fundamental about the basic machinery of the cell.
Exactly. It suggests its pre-training on huge amounts of data gave it some meaningful biological priors about these highly conserved, co-regulated systems.
That's a huge step forward. It's not just guessing cell cycle arrest anymore. It's showing some understanding of protein synthesis.
And they took it one step further. They tested its downstream utility. They asked if the model's predictions could distinguish between perturbations that cause low versus high chromosomal instability, that CIN phenotype.
So using the prediction to forecast a real measurable outcome.
Yes. And again, only the fine-tuned scGPT performed substantially better than chance with an AUC of 0.73. It proved it was retaining enough specific biological information to partially predict a complex, unseen phenotype.
So this deep dive really confirms that the field has been maybe a little misled by its own data and its own metrics. Systema feels like a necessary course correction.
It absolutely is. The big takeaway is that systematic variation is a pervasive, universal issue, and Systema is the tool we need to see past it and focus on the real, specific biological signals.
So looking forward, what do we need to do to finally build models that can actually deliver on that promise of revolutionizing therapeutic discovery?
Two big things. First, on the data side, we need larger data sets with highly heterogeneous gene panels. We need to intentionally design screens that target unrelated genes to dilute that systematic noise from the start.
And second.
Second, our evaluation has to shift permanently towards biological utility. We need to stop obsessing over abstract correlation scores and start asking how well our predictions help us answer real questions about cell behavior, morphology, or drug response.
So here's where it gets really interesting for you. If removing all these biases drops the performance score so low, what is that essential kernel of biological knowledge? Is it protein interactions, regulatory motifs? What is the fundamental thing that the next generation of models has to internalize to finally move beyond these simple averages? Go explore the idea of Systema and let us know what stands out to you.
Until next time, keep digging deeper.